Good afternoon, everyone. This is my very first visit to Oxford, and I'm having a grand time so far. I'd like to start by thanking John Keating and the entire event staff for the invitation and for getting me here. And it really is my pleasure to be able to talk to you a little bit this afternoon about why I think it's important for mathematicians and quantitative and people who do quantitative science to have a seat at the table when the conversation is about cancer. So the very first half of my talk is really geared towards the students in the audience. What I'd like to hopefully do is convince you when you leave here about the untapped power and potential of mathematics to bring to bear new tools on the fight against cancer. So I wanted to start with this Time magazine article that appeared a few years back. And the tagline of the article was, "The hero scientist who defeats cancer will likely never exist." And then they went on to describe cancer in this way. Cancer is an intricate and potentially lethal collaboration of genes gone awry, growth inhibitors gone missing, of hormones and epigenomes changing, and rogue cells breaking free. It works as one great armed force attacking by the equivalent of air, land, and sea in stealth. And basically, what are we going to do about it, right? This article said there will not be one exalted individual. There's not going to be a victory celebration. No Marie Curie, no Jonas Salk, who in 1955, after he created that first polio vaccine, was asked, "So what are you going to solve next? Cancer?" It's really not how these complex diseases work, right? One person can't just move from disease to disease and find an answer. So there very likely won't be one hero. But there may be many heroes, right? So this article went on to talk about all of the kinds of expertise that might need to be brought to the table to help fight cancer. So they, of course, had biochemists, molecular biologists, biomedical engineers, geneticists, pathologists, biostatisticians, they even mentioned, informaticists and medical oncologists. So that's a lot of people. But you know who they didn't have? They didn't have mathematical modelers, so I had to draw myself and us in there. Because again, what am I going to do today? I'm going to convince you that we deserve a seat at the table. So all of this kinds of expertise needs to come together in a collaborative effort to address the challenges associated with cancer. Right, and so this is more of a team-based science where we can all have complementary places and tools that we bring to bear. Why do we need this? Well, cancer is a complex disease. It's actually a single name for over 300 diseases that are genetically different, and so we have to take that into consideration when we're thinking about how to treat the many diseases that are cancer. Not to say that cancers don't have some common features, right? So most cancers start with mutations acquiring and accumulating in our normal cells, and they go through similar processes of progression. So a small tumor might form. That tumor's not getting enough nutrients from the surrounding tissue, so cancer cells initiate new vascular network formation, connecting the cancer to the highway of blood vessels in our entire body. These cells can enter the bloodstream and move around and start new tumors in other places, which is what makes cancer so dangerous. So given all of this, all these different stages, the complexity of cancer, I argue that we really need the field, not just one person doing this, but the field of mathematical oncology. And researchers with mathematical backgrounds have been using mathematical tools and approaches to understand better how tumors grow and to predict how best to treat them for over a century. This is not new, right? Today, mathematical and computational approaches have been used to address every single stage in the cancer progression cycle and at very different scales of biology. We'll talk about multiple scales in just a minute. But mathematical oncology is bringing those tools to the table. This quote kind of lets you know how the field has kind of evolved, right? It's been around for a long time. It originated as a subdiscipline of maybe applied mathematics, right? But now, today, it's even being considered a subdiscipline of oncology itself. And that shift is critical. That shift in thinking to where mathematical tools are accepted, an accepted part of medical oncology research is exactly what we need to happen to use all of the potential of math against these kinds of problems. So why should we worry about or attempt to mathematically model cancer? There's a number of reasons, but one is to help reveal possible, potentially hidden underlying mechanisms of the dynamics that are contributing to cancer progression. Right? Experiments are necessary and will always be necessary, but often they're not able to measure all of the key, most important, critical quantities of interest. They can get close, but maybe not all of those. So, mechanistic mathematical models of the underlying dynamics can help shed light on the unknown. Right? So that's one reason that we try to use mathematics in cancer. Another reason is that experiments often isolate down to a very specific question, right, or a specific aspect of a system like cancer. And so that might get you a really great answer to one part of a very large question, right? So we might have a fragmented understanding. What mathematical models can do is integrate results from a variety of sources, from a variety of scales, providing essential additional information that can kind of fill in the gaps, right? So mathematical models can help solve that piecewise jigsaw puzzle and get us maybe to a fuller understanding or a more complete picture of what's happening. So those are some of the reasons that we choose to spend our day jobs trying to develop mathematical models of cancer. Beyond that, I really want to argue that there is a need for this discipline. And one reason for this need is more data, right? More data leads naturally to the need for more math. We are in a data gold rush right now. The volume, the amount of data, the number of different sources of where we get data is growing exponentially, right? And these data-rich information sets are hard to understand without analytical tools. As one example, what you're looking at up here is a cell, right? And its cell membrane has all kinds of receptors and gated channels, ion channels that allow the cell to experience its environment, right? So when the cell is trying to synthesize all of this information from around its environment, that information is cascaded through very complex signaling networks. There are feed-forward loops, feedback loops, there are redundancies, there are checks and balances. And using mathematical modeling of these signaling networks can help integrate the experimental knowledge that we have about particular pathways within the cell, integrate that knowledge into a coherent picture that can be used to generate hypotheses about how cells make decisions. So they get all this information from outside. How do they know when to divide, when to die, when to move, when to differentiate, right? How do they make that decision? Integrating all of that into a mathematical picture can help answer questions like that. Another reason for the need for math associated with data is we're just getting, technologies are providing big data sets, right? So this is an example of the Cancer Genome Atlas. So that atlas produced 2.5 petabytes of data. Just to put that in scale, that would be over 200,000 CDs of information on 33 tumor types, including rare cancers, from 11,000 patients with and several different types of data at different scales of the cancer problem. So we've got all of this data, right? What do we need? We need time and cost-efficient strategies to analyze it, to help understand it and to interpret it, right? And to extract meaningful information from it. So how can we do that? We can do it with math, right? We specifically need mathematical approaches to generate reasonable testable hypotheses from all of this data. And you could go back to some traditional classical mathematical approaches, for example, like a principal component analysis, which just makes sense of these big data sets by taking their large number of variables and reducing them down to the principal components that explain the behavior. So that's kind of an old traditional method. But newer methods are emerging, like topological methods. So topological data analysis really has potential to play a big role in understanding these huge data sets like the Cancer Genome Atlas. All right, so more data, more math. More math has led to new ways of thinking about how to use mathematics. All right, so here we talked about these large data sets. They can be used to guide mechanistic models, right? And these mechanistic models kind of capture what's going on in our system at different levels of resolution, right? And those can be combined with, a separate set of validation experiments to generate virtual cohorts of patients that can then be used to test new therapies or new technologies through in silico or experiments that are done computationally instead of on actual patients or mice. So more math has led to new ways of thinking about how to use it. How can we use math to really push forward the field, right? So new math has led to the need for new computational power. And advances in computing power is enabling us to do calculations and simulations that just could not be performed years ago. So this slide is sort of a prediction of computational cost for a computational cost for a personal computer that would have the processing power of a human brain. The prediction in 2001 was that we would have such a thing by 2020. For $1,000, your personal computer would have the processing power of a human brain, right? And that prediction went further to say that by 2030, it would take a village of human brains to match a $1,000 personal computer. So where are we? Are we there yet? Not quite. The latest and greatest supercomputers can do a quintillion of calculations per second. Our brains are a miracle. We can do better with our brain still, right? But there is a supercomputer that's scheduled to go online here in just a couple months that will actually rival the processing power of a human brain. So we're getting there. We're getting there. It's coming this year. So what do these new computations get us? What do we get when we're able to do computations like this really fast? New computations lead to new possibilities, right? So the possibilities for us to perform experiments using the computers are essential because it's expensive and it takes a long time, and we need to be humane about how we conduct experiments on animals and humans, right? So this is a picture of some research from the University of Texas, Austin, who's using these researchers are using a supercomputer to help look at cancer growth in a brain tissue slice before and after treatment, and they can get down to the details of the spread of the cancer and visualize it in layers of brain tissue. And predict effective therapies. So that's one example of new computational power using these supercomputers, leading to new possibilities. Here's another example. So here, what are you seeing? You're seeing a blood vessel network infiltrating a tumor. Think about it. There are over 60,000 miles of blood vessels in our body, right? They carry nutrients to make sure our tissues are oxygenated. But when it comes to cancer, they can also carry cancer cells to all of these other parts of our body. So a supercomputer is helping a team of researchers at Duke University to predict the spread of cancer cells in that complex, tortuous kind of vascular network to predict where metastases might occur and when and how. So that's just another example of new computation leading to new possibilities. So clearly, there is a need for this field called mathematical oncology, right? Because cancer is a dynamical system. And it cuts across all these layers of biological complexity. Mathematical tools are the natural choice, right, for understanding dynamical systems and multi-scale processes. So what this slide is showing are all the levels of biological organization that you can think about when you're asking questions about cancer, down to single molecules, to proteins, to single cells that affect the whole cell population and eventually affect organ systems and whole organisms. So mathematically speaking, researchers have been using different kinds of mathematics to address questions at all of these different scales. And depending on what your question is, what your biological question is, that may inform your choice of mathematical model or mathematical tool, right? But notice that these regions are blurred, the boundaries are blurred a little bit, because that's what's happening now, today. We're not just asking questions at one scale. We want an integrated knowledge across these scales, which means we need integrated mathematics across these scales. So the work that I do, and then I'm going to get to talking to you about today, looks at questions that start at about this level, those signaling networks inside cells that we talked about, that leads cancer cells to make the wrong decisions that affect the entire cell population and cause a growing tumor. And we use several different kinds of mathematics to answer those questions. So let's get into some of that, right? So what I want to talk to you about today is some of the work that I'm doing looking at therapeutic options for treating cancers. And we're going to look at two different kinds, and we're going to look at an immunotherapy, and I'll talk about that, and then a molecular targeted therapy and how we combine them and how mathematics is helping us to understand better ways of doing that. So the actual goals of this particular research project is to combine multi-scale ordinary differential equation and agent-based models, and I'll talk about what each of those kinds of models are. We're combining those mathematical modeling approaches with experimental technologies that quantify the cellular dynamics at the cellular scale, so we can get actual measurements of what cells are doing when they interact with each other. And we want to do this for two reasons. The first thing we want to do is see if we can't develop predictive methods for improving the effectiveness of these two types of therapy, immunotherapy and a novel therapy that actually targets cancer-driving mutations on cancer cells, right? And the second thing we want to do is see if we can't gain a more robust, basically just a deeper understanding of how specific tumor mutations affect the immune system and can impact therapy, either making it worse or maybe improving it. So those are the things we want to do. So the mathematical models that I and my team are developing are flexible and general enough to be applied to a wide range of solid tumors and combination therapy types. But I'm talking to you today about some particular work that I'm doing with colleagues who are medical oncologists at the University of Chicago. And they're working on bladder cancer, which is one of the 10 most common cancers worldwide. It's not getting any better. In fact, its prevalence is increasing globally. And honestly, we're just not doing a good job with this cancer, right? So when it gets to its advanced stages, the 5-year survival rates are low. And the cornerstone of treatment, the best we got, is not good enough. It's not working on this cancer, and things just aren't getting better. So what do you do when the hallmark of medical treatment for the type of cancer that you have, in this case it's cytotoxic chemotherapy, what do you do when it fails? When it is failing miserably and it's not getting better and it hasn't gotten better for decades? What do you do? One answer might be targeted therapy. So targeted cancer therapeutics are drugs designed to interfere with specific cells or molecules or enzymes or proteins that the biology tells us are key drivers of that particular type of tumor's growth. A primary goal of this more targeted therapy is to reduce side effects, right? To fight cancer with more precision. Instead of giving a drug that just blanketly kills cells that are dividing, let's kill specific cells. So that's the goal of this targeted therapy. So maybe this could be an answer in bladder cancer as well. So what could our target be? What should we target? Well, here's where we go to those big data sets, and here genomic analysis has identified frequent alterations and mutations in a particular family of proteins. These mutations lead these cells to divide more and to live longer, right? So this could be a particular target. If we can stop these mutations from giving these fitness advantages to these cells, that could be an improvement. Okay? And there have been some clinical trials, some very limited trials on drugs that do just this. So drugs that stop these these cancer-driving mutations are showing some promise. In some cases, it's just really hard to predict what patients are going to respond to this. But they have approved in the States the very first targeted therapies of this type. So that's showing some promise. We just need some help to figure out how we can do better with it. So remember I said we're gonna try to mathematically model two different types of therapy. The second kind of therapy is immunotherapy. And that is arming your own body's immune system to better fight cancer. So our immune system should be able to do a better job than it is doing. And in fact, this type of therapy, immunotherapy, is just really dominating the media right now. You see words like it's a new weapon, it's a game changer, it might offer a potential cure, it's a breakthrough for the biggest killers, right? So this is important. Maybe this is the answer. The best we got is failing, so maybe this is a new direction that we should try. It turns out that bladder cancer cells can actually suppress the body's immune system. And so when you give these drugs, you can actually stop this suppression and give the immune system the boost it needs, right? But again, the response isn't great, and it's hard to predict which patients will respond, right? I wish that were the end of the story. It's not. The plot actually thickens. These cancer cells have mutations that give them a fitness advantage, and those mutations are blocking immunotherapy from working. So what you're seeing here, time and days and how big a tumor is, when cells don't have a mutation, they grow, as you might imagine. If you give immunotherapy, you get a significant reduction. The therapy sort of works. That same scenario, when cells have this mutation, cells grow when you don't treat them, and cells grow when you do treat them. So that mutation is doing something to impact the effectiveness of immunotherapy. You've been patient, you've waited. We're going to start talking some math. I had to tell you the biology story though. So it could be, so this is not doing too well, that's not doing too well, cornerstone of treatment isn't doing too well. There may be a way to find some synergy if we had a co-acting combination of this very potent immunotherapy that's boosting our body to do a better job, and this very targeted mutation-specific therapy may be able to do what the cornerstone of treatment, chemotherapy, couldn't do. All right, so here's where we start thinking about, can a mathematical model help us understand how to do this better? One more piece of data before we get there. So they've tried this. They've tried co-treating with an immunotherapy and a targeted therapy. This is showing the percent change in the tumor's volume after treatment for 23 patients. Notice some patients, it was like you didn't treat them at all. Their tumor volume increased significantly after treatment. Others still had 20 to 30% increases in tumor volume. And then some patients actually responded. And lower doses seem to make the responses better. This is just so confusing to the medical oncologist. What do I do? I thought this would work. It's not working. And when it does work, I don't understand why it's working, right? So what do you do when the going gets tough, when what you thought would work doesn't work? Well, it depends on who you are. Are you a mathematician or are you not, right? So we know what a mathematician would do. We know what a mathematician would do. Mathematician would turn to the power of mathematics, right? So using a data-driven mathematical model is a powerful and practical way to help figure out how to increase the efficacy of these kinds of drug treatments, right? We're attempting to combine two very different therapeutic options, right, that have very different mechanisms of action. The experiments show us that it's a mess. We don't know when and where and how it's working, right? So maybe mathematics can help tease out some of these things. Okay, so I want to talk to you about two separate mathematical models. The first is going to be an ordinary differential equation model designed to answer specific questions about the impact of these mutations on tumor growth and therapy. Okay, so I want to use the model to answer some very specific questions. Question number one, under what circumstances will the immune therapy outperform the targeted therapy if you give them by themselves? So as single options, are there people, patients, options where you would expect one therapy to outperform the other? That's the first question. The second question is, how will a virtual cohort, a computational cohort of patients with different mutation-induced fitness advantages, so different amounts of increased proliferation and survival, respond to therapy? And the third question is, can different dosing strategies improve combination therapy? So let's see if we can develop a model that can address some of these things. So here's a schematic of the model. Before I show you any equations, let's just show you what the key components are. We've got some immune cells in the model. We've got some tumor cells in the model, right? So that's the cellular scale. But on these tumor cells, there are these mutations in proteins and receptors that cause the cells to proliferate too much and to live too long, right? And so we're going to give a drug that blocks all of this. So we see some X marks in there to block all of that extra fitness advantage. And then at the same time, we've got these immune cells that try to come online to attack the tumor cells, but the tumor suppresses them unless we give an antibody that blocks that suppression and gives the immune system that boost. So those are the key features of the mathematical model. And here's what the mathematical model looks like in this case. So we have two different scales that you saw me just talk about. We had the cells, so we have tumor cells and we have immune cells, but then we had all of those molecular species that were on the cell surfaces that are the mutations that drive these fitness advantages. So we have to write down equations for how the tumor and immune cell populations change, but how those changes are driven by this molecular level detail of those mutated receptors. And then we have a nice linker of our two scales. So this is exactly what I mean about multi-scale modeling. We have things happening at the cell level, we have things happening at the molecular level, and then we link them together to make sure that those molecular details are driving those increases. Okay, so I'll leave that there for the math. All right, so what kind of analysis and questions can we ask of the model just to understand the model before answering those biological questions? Well, one thing we'd like to understand about the model before answering any biological questions is, which parameters most significantly influence how the tumor is going to grow. So we do a sensitivity analysis. Here we did a global analysis using partial rank correlation coefficients, and we found that of all the parameters in that model, 4 were the most sensitive, caused the biggest changes in the tumor volume. And they're probably the ones that intuitively you would think would be important. So how fast the tumor cells divide and how often they die, those are the first 2 rates that are really important. And then that fitness advantage that you get from the mutation on both of those phenomena. So the fitness advantage associated with mutation giving proliferative advantage and survival advantage turn out to be the 4 most important parameters. Since those 4 have the biggest effect on how the tumor grows, we want to make sure we estimate them with the greatest precision. We want to do the best job making sure we have good estimates for them. So we have some data. I showed you data before of what happens when you don't have the mutation and how tumors grow. So you can fit the model parameters associated with the baseline proliferation and survival to that data. And then we also have some data for how the mutation grows. So we can figure out what the fitness advantage actually is by fitting to this data set. Okay? So that's calibrating the model, right? But we're also going to be giving some treatment. So we have one more calibration step. We fit one additional parameter to a treatment data set. Now we're giving that immunotherapy using the regular dosing schedule. And we estimate the binding rate of the drug, or the antibody. And then we're done with calibrating the model. We want to see if we can validate this model and make sure this model would predict exactly what experimental data would predict. So we have an entirely separate data set for what happens when you have the mutation and you give this therapy. And we simply simulate our model and overlay it with that data. And we see that our model captures the average trend really, really nicely. No data fitting here. This is our validation step. So we know that our model is doing kind of what we hoped it would do with prediction, with predictive power. So now we can start answering some questions. The first one was, under what circumstances will immunotherapy outperform the targeted therapy? So let me walk you through the answer to that question. What you're seeing here is a heat map showing the difference between the outcome of immunotherapy and targeted therapy on day 25, which is kind of the last day of the experiment in mouse. What you see up here in green, so when you have these lighter green colors, that means targeted therapy is doing better. When you have these darker blue colors, that means immunotherapy is doing better. So let's just start up here, say like in region 3. What does this mean? On the x-axis, we have the impact of the mutation on proliferation. And on the y-axis, we have the impact of the mutation on survival. So these are those fitness advantages, right? So if we have large fitness advantages for both division and survival, right, then this is suggesting that we should give targeted therapy by itself. It will do a great job all on its own, right? And if you had the other situation where there were almost no fitness advantages, then of course you might not want to target anything because the mutation isn't driving the results. Right? So you might want to stick here where immunotherapy is better. But what is interesting is that if you start anywhere over here in this green region where we have high fitness associated with proliferation and you reduce that fitness, you eventually move into a region where immunotherapy does better. So moving from right to left, we go from green to blue, right? But the opposite isn't true. If I start up here where I have a high fitness advantage associated with survival and I reduce it, you know, I never get into a region where blue turns to green by doing that. Right? So the proliferation impact determines the best therapy, not necessarily the fitness advantage associated with survival, is what we found. Okay, second question was, how will a virtual cohort of computational patients with different mutational fitnesses respond to therapy? So in order to answer this question, we have to generate a virtual cohort. So how are we going to do that? Well, we're going to ask a question about how reliable were those fits that I showed you of the model to the data? Well, it turns out not all of our sensitive parameters are uniquely identifiable with that data. So what that means is there's a range of parameters that could fit that data equally well. That happens all the time with noisy experimental data and these, simplified mathematical models. So it's not surprising. But what we can do is exploit that. Instead of thinking of it as a negative, let's turn it into a positive and give a prior distribution on those parameter values, and then use your favorite method. We used MC with Hastings sampling, so Markov chain Monte Carlo with Hastings to come up with a posterior distribution of what parameters, range of parameters will fit that data the best, right? And we use that, all of those parameters will fit the data, so that's a virtual cohort of heterogeneous patients that all grow the same before treatment. Question is, how do they respond to treatment? So we can use that as our virtual cohort to test these different strategies on. Okay, so here's an example of some outcome. You have 50 virtual patients. We're giving them both therapies at the same time. This is what the experimentalists tried first, was just co-treating with each kind of therapy on their regular schedules. And we see what happens if you don't treat at all in blue. Almost none of the patients survive, only about 14% survive to day 25. And then if you give each therapy on its own, we can improve that. But then if you give combination therapy, which is up here in green, we can do even better, getting up to about 86% surviving of that virtual cohort of each patient having different fitnesses. About 86% of them will survive with a combination therapy. Third question was, can different dosing strategies improve combination therapy? So when they had the brilliant idea of combining these two kinds of therapies, they said, let's just give them both together at the same time, right? So maybe that wasn't the best way of doing it. Maybe there's some other ways to do it. So we just did a quick check to see if you co-treat versus if you pre-treat with one drug or the other drug, do you get better responses? So instead of co-treating, maybe you give the targeted drug first, or maybe you give the immunotherapy first and see what happens. So this shows no therapy, how big the tumor would be, and then these show the reduction in tumor size based on the other two schedules. So we can cross out schedule number 2 immediately. We don't get much reduction. And schedule number 2 was pre-treating with our targeted therapy. That does not seem to work. But the other two are relatively close, 27% reduction versus 31% reduction by day 25. But my headline up there says that you should choose this one, that pre-treating with immunotherapy is the better therapeutic option. Where did I get that from? 27 is definitely less than 31, right? So why would I say that this is the better therapeutic option? Well, let's compare this to this, right? Look at how many fewer doses of drug I needed to get a similar amount of reduction, right? So I gave 15 doses here of the targeted and 6 here. I only gave 10 here and 4 of the other. So I'm saving money because these are very expensive therapies and side effects associated with giving 2 drugs at the same, 2 drugs in a regimen. And I'm still getting a relatively similar amount of reduction. So medically speaking, my collaborators tell me that they would always choose this option. Less drug, less hardship on the patient, and similar reduction to therapy. Okay. All right, so we have used our model to answer, one type of mathematical model to answer several questions about therapy. Now let's use another type of mathematical model to ask some different questions and answer some different questions. Now our focus is going to be the impact of cancer mutations on the immune system. All right, so first it was just impact on how the tumor is going to grow and how it's going to respond to therapy. Now we're going to ask a different question. How do different assumptions about how the mutation impacts the immune system affect the outcome of therapy? So they don't know. They don't know exactly what the mutation is doing to the immune system, right? But they do know, again, here's an example of using those big data sets from the TCGA. What they do know, what they did was look at all the data in the TCGA and found that there were 3 distinct subtypes of this particular kinds of cancer. Ones with low, medium, and high levels of immune cells present, right? So they call them inflamed, non-inflamed, and then some middle ground, right? So hot tumor, meaning lots of immune cells, cold tumor, meaning not so many immune cells, and then some middle ground, right? But then they went one step further with this data and found that when you had cold tumors that were not inflamed with or did not have a lot of immune cells, they all were mutated. They all had the mutated cells. And that kind of tracks with what we saw earlier, right? When you give the drug and there's no mutation, we get a distinct effect. When you give the drug and there is a mutation, immune therapy has no effect. So that we know that the mutation is doing something. We just don't know what it is. So we want to use a different kind of mathematical modeling to try to answer some questions about this. So this is an agent-based modeling framework where each individual cell in our system has independent decision-making power, and it makes decisions for itself. And we represent these on, in this case, we're using a three-dimensional lattice where space is partitioned into voxels, and we have cells in each voxel. We start the simulation with just a small number of tumor cells in the middle and no immune cells. Immune cells are recruited to the tumor site, and we update in time. Okay, then we give therapies according to just how we did it with the other model. And then the cells are able to interact in time and in space now. It's the big difference. So the spatial model, the other was just temporal. So in space, they interact with each other, with the drugs, and with other signaling molecules in the environment. So in this model, we have several different kinds, we have two different kinds of tumor cells, the ones that attract immune cells well and the ones that don't. We have different ways that immune cells kill, and then some different states of the immune cells. And this kind of modeling is really nice because you can do a 2D tissue slice and get something that looks like a histology of what the cancer might look like. You can do 3D simulations where you just let it grow and you can see what's going on in 3 dimensions. But what we wanted to use the question for, and we're almost done, is to use this model for was to test hypotheses about what the mutation is doing to the immune system. So we are hypothesizing or testing 2 different hypotheses. Maybe the mutation affects recruitment, so the way in which immune cells come to the tumor site, right? Or maybe the mutation affects the killing, right? So maybe cells who have this mutation, the immune system just can't kill them as well. So we might have 0, none of those effects. We could just have the cytotoxic effect. We could just have the recruitment effect. Or it could be that both of these things are happening at the same time. So we want to see what might happen. So when we run the simulations of combination treatment, when the mutation has no impact on the immune system, we get that the tumor is completely cleared. So all that's left are deactivated immune cells, our tissue looks nice and clean. And when we run the simulations with both the recruitment effect and the cytotoxic effect active, we get large, fully formed tumors where every cell type is persistent in the population. So what's interesting about this is, let's see, do I do this here? If you watch these movies play, so this is a case where we get elimination, and this is a case where we get escape. Right? So I said this is where we get elimination. Look how big this tumor is. Right? So this tumor is getting huge. But watch what happens. And this one's escaping. Right? It's growing, growing, growing. And lo and behold, eventually it's cleared. If I just would have looked at that one temporal picture of the tumor being gone, I never would have known that that happened. That tumor got huge. And then the body was able to control it. This one escaped, and the immune system was never really able to control it. So what's the difference here? The combination therapy can cure a hard to treat tumor if the mutation doesn't affect the immune system. And combination cannot clear an easy to treat tumor if the mutation does impact the immune system. It's kind of the take home message there. All right, let's go one step further, last thing here. So which one of those effects is the most important? So again, we have these hypotheses for the effect of the immune system. Maybe it's inhibiting recruitment. Maybe it's making these cells resistant to killing. So here, purple represents neither one of those things happening. Blue represents both of those things happening. And then our diagonals are either the cytotoxic effect or the recruitment effect. So let's just look at what happens when the tumor is cleared, when the tumor size goes to 0. In both cases, we're in purple and red, right? So the mutation did not inhibit recruitment, right? In these 2 cases, yellow and blue, recruitment is inhibited, and in all of those cases, in both of those cases, the tumor was able to grow. So when the mutation does not affect recruitment, the tumors respond to therapy. So this effect is the biggest effect, the recruitment effect. When it does inhibit, that's when we get this escape. Okay, I think I'm going to wrap up here. I'll skip that one. I think we're just about out of time. So, summarizing. I hope I've done what I set out to do for the students in the audience, which was just to convince you that there is untapped potential and there's real power in using mathematics and quantitative approaches to try to help understand certain aspects of how cancers work, how they grow, and how to treat them. There's also this translational value, right? These kinds of experimental and computational studies that I showed today can lead to generating new hypotheses, like recruitment is the driver, right? So a new hypothesis regarding how to optimize or how to target or to do better with treating cancer, right? So platforms like this that use mathematics are flexible, they're robust, they can be applied across a wide range of tumor types, right? It's really how you connect it to data that makes it specific to a cancer. So you're really developing tools that can be used broadly when you do this. So my take-home message is just do the math. All right.